I'd like to welcome everybody to the April 16th meeting of GLIMPRINT mini seminar series. Appreciate you for coming today. We have one talk today by James Doran from University of Bath, model of tuberculosis progression using CompuCell 3D. As always, I need to remind people that this meeting is live streamed and recorded and available publicly in edited form later. Tom Helikar, myself, Jim Sluka, Lorenzo, and Ellen are always available to take your comments and suggestions. Let us know what we can do to make things work better for the seminar series and for the community at large. We have a variety of communication channels that work better if they're used. We have the IMAG MSM wiki page. We have the updated GLIMPRINT page. We have our YouTube channel, LinkedIn channel. We appreciate your help getting the word out and building community. If you have a publication to share, that could be shared on the IMAG wiki page and also on the GLIMPRINT page. Let us know. Email Jim Sluka, jsluka@iu.edu if you want to do that. And we'll do our best to get that up and that information out. If you have information of other kinds, seminars, meetings, workshops, or classes, job openings, funding, announcements, those can be put up on the GLIMPRINT page as well. You can reach out to jsluka@iu.edu for help on that. We're happy to get your information out, and we'd love feedback on the new GLIMPRINT page. On April 30th, we have Ron Kennett speaking. On May 14th, Kevin Flores and Carolina Gonzalez-Cervasos will be talking on May 28th. We always appreciate your suggestions for speakers. Think broadly. We can have yourself, your students, your postdocs, people who've written papers that you're interested in, anything that you think might be relevant to the broad area of multiscale modeling, medical digital twins, infection, and immune response. Those could be experimental, those could be computational, they could be background methodological, they could be data analysis, it could be drug development, treatment development. Think broadly. Again, this forum is one for people of all backgrounds and interests related to multiscale modeling of immune response. So please help us fill the schedule. Coming up, we have modeling the invisible, competition on forecasting viral spread with limited data. That will be in person, June 3rd to Saturday, June 6th, at Georgia State University at Atlanta. That's being led by Jim Sluka, Yi Zhang, and others in this community. It's especially relevant to students and postdocs, but it's open to everyone. It's an exciting program. Could be a lot of fun, and I hope that people will consider attending. We also have the announcement now for our CompuCell 3D workshop for this summer. That's going to be a virtual workshop from July 27th through August 9th. It's going to include a week of basic CompuCell. If you already know CompuCell, there's going to be a week of advanced CompuCell, and then there's a 3-day hackathon where people can try out and build ideas using the software. We'd be delighted for people to participate. We will have a steering committee meeting tomorrow led by Tom Helikar. It's going to meet a half an hour later than usual from 11:30 to 12:30 Eastern Daylight Time. Please attend. If you think you should be invited and you haven't received an invitation, reach out to Lorenzo by email or in the chat, and he can add you to the list. We always appreciate the help organizing this community. Okay, we'll move on to our speaker today. We're very pleased that James Doran has agreed to speak to us on modeling tuberculosis progression using CompuCell 3D. This is a topic of great interest in infectious disease and in treatment, and we'll hear about his approach to modeling this complex problem.
Great. So hi everyone. Yep, my name is James Doran. I'm a final year PhD student at the University of Bath in the United Kingdom, and in collaboration with my fellow co-authors listed here, Christopher Rowlatt was recently a postdoc at the University of Bath, Jibin Powerthill from Swansea University, and my PhD supervisors, Ruth Bonus and Christian Yates, also from the University of Bath. We have developed a within-host model of tuberculosis infection with the CompuCell 3D software. So to give a brief outline in today's talk, I'll give some context on tuberculosis, the seriousness of it, and the interesting quirks that make it an interesting disease to model. I'll describe the bacterial, the biological mechanics of infection with the causative pathogen, and how we've tried to accurately model this using CompuCell 3D. We'll then present the results of our model and compare them to a similar agent-based model that had been previously developed to investigate TB within host dynamics, and then discuss the implications of our findings and suggest some potential future work. So tuberculosis, or TB, as I'll typically shorten it to for the rest of this talk, is an airborne disease caused by inhalation of the pathogen Mycobacterium tuberculosis, or shortened hereafter to MTB. It's typically expelled after any respiratory activity via aerosols, and it can then be inhaled by susceptible individuals. Although TB infection can occur in any part of the body, typically it usually occurs in the lungs. So I'm mainly focusing on pulmonary tuberculosis for the rest of this talk. Aside from the 3 years from 2020 to 2022, when COVID-19 was the leading cause of death from an infectious agent, tuberculosis typically holds this title. In 2023, it is estimated to have caused 1.25 million deaths worldwide. Despite this, it is a disease that impacts people in a lot of different ways. It's typically estimated that about 1 in 4 individuals globally has the MTB pathogen within them that could cause active disease. However, about 9 in every 10 individuals will go on to have a latent infection and not get sick. Only about 10% will actually develop active TB and require antibiotics, which is typically 6 months of antibiotics in order to cure the disease. And as I say, without antibiotics treatment, the risk of death is clearly quite high. As you would imagine, for such an important infectious disease, a lot of mathematical modeling has been conducted to try and investigate how we can improve the situation, improve treatment outcomes, etc. One recent bit of modeling work was conducted by Bonus et al. In 2018, where they developed an agent-based model, and they found that the spatial organization impacts whether or not the outbreak will be contained and a latent infection will result or not. So in particular, the position of the initial bacterial cluster of MTB within the lung tissue and the spatial arrangement around it of blood vessels and other immune cells was found to be important over the course of the 12,000 simulated hours or 500 simulated days in determining whether or not the infection would go on to be latent or lead to active disease. So to try and provide some cross-validation for these results, we've developed a similar model using CompuCell 3D. For anyone unfamiliar, this is a software that has been used to model a range of biological cellular phenomena, including cell sorting, tumor growth, and with regards to infectious disease, has also been used for COVID-19. It combines a cellular POTS model, also commonly known as the Glaser-Grainer-Hogewerg model, with partial differential equation solvers for chemical fields, and also gives the ability to, for example, add in intracellular processes as well. We believe that this is the first TB model that has been developed using CompuCell 3D, and we've used this to investigate the first 200 hours post-infection. So I'll briefly summarize the biology of MTB infection in this slide. It's worth noting that, of course, our model is inevitably going to be a simplification of the real-life biology and the real-life interactions between pathogen and host. In particular, we've only considered interactions that were incorporated into the original model presented by Bonus et al. In 2018, which is known as the within-host infectious disease model, and hereafter I will just shorten it to WHIDM. As an example of ways that simplification has been made, it's well known that a range of different cytokines are known to be important to TB within host dynamics, but our model will consider just one, which is a generic chemokine used to signal chemotaxis. It's been observed that MTB pathogens, MTB bacteria will replicate at different rates depending on their phenotype. And in particular, it has been observed that fast-growing extracellular bacteria will enter a dormant state and reduce their replication rates if they find that the environment they are in is not particularly hospitable to replication. So one such factor may well be hypoxia or the lack of sufficiently high oxygen in their local vicinity. So in our model in particular, we have made MTB replication rates dependent on the local oxygen concentrations to try and capture this phenomenon. In our model, we've included a few different immune cells that are typically observed within the immune response to MTB infection. And our immune cells reach their site of infection by following chemokine gradients through the process of chemotaxis, where if they sense larger chemoattractant concentrations, they will move closer towards those. In general, the immune response to tuberculosis can be summarized as being in two stages. There's the initial innate immune response and the subsequent adaptive immune response. As we're only looking at the first 200 hours, it's worth noting that we are mainly focusing on the innate immune response in this talk. So when the MTB bacteria first arrive in the lungs, the first immune cells to encounter them are often macrophages. These are an immune cell which tries to phagocytose the MTB, or in other words, they try to ingest the pathogen effectively and destroy them from within. However, MTB has been with us for a long time and has evolved to resist these efforts. And so although it will be phagocytosed by the macrophages, it will continue to survive. It will enter this dormant state, and subsequently the macrophage itself will become infected. At this point, they will tend to secrete chemokines, warning other nearby immune cells that there is an infection and signaling them to travel towards this site of infection. And if the macrophages continue to phagocytose too many more MTB, then eventually they reach their carrying capacity, they burst, and their intracellular bacterial load can then be released back into the lung tissue. The second stage is then the adaptive immune response. Cells such as dendritic cells will typically take information away from the site of infection to the lymph nodes, which will then be used to activate T cells. These will then travel back to the site of infection, and although T cells come in a wide variety of different phenotypes and perform a wide variety of different functions, their main two functions with regard to the response to tuberculosis are broadly twofold. Firstly, they help activate macrophages. This means that macrophages can phagocytose MTB without having such a restricted carrying capacity, and it won't cause them to burst if they continue to phagocytose too many, and helps them to actually destroy the bacteria. And the second main aspect of T cells response is to try and help kill infected macrophages, and then subsequently also kill the phagocytosed bacteria within them. As we're only looking at the first 200 hours, this adaptive immune response hasn't ultimately been included in our final CC3D model, but it's worth knowing nonetheless. The reason we've chosen not to include it is because typically T cells don't actually reach the site of infection until about 9 days after the initial infection occurred. And as we're only looking at the first 200 hours, this time period, we wouldn't expect to observe any T cells within the infected lung tissue. Usually, as a result of all these within host dynamics, we wind up typically with one of two outcomes. Normally, for most people, again, about 9 in every 10, the outbreak will be contained in a structure known as a granuloma. This is an aggregation of immune cells, bacteria, and necrotic tissue. And if it's a small, solid, well-formed structure, the bacteria is effectively walled in and can't disseminate further through the lungs. If the granuloma does not form effectively, however, the bacteria can disseminate, spread throughout the lung tissue, continues to replicate, and leads to active disease, which requires antibiotics treatment. So now I've described the biology of it, I will now go into how our CompuCell 3D model looks to capture all of this. The schematic on the right gives a brief idea of how the different parts interact with each other within our model, but I'll describe each of these in a bit more detail in the following slides. And it's worth noting that if you'd like further information on CompuCell 3D, then a good starting point would be this reference I've included at the bottom of the slide, written by Swat et al. In 2012, which serves as a good starting point in terms of how CompuCell 3D works and what it can be used for. The screenshot on the right shows the initial conditions of one of our simulations. In our model, we have looked to model a 2 millimeter by 2 millimeter cross section of lung tissue within this square boundary. We've chosen this size specifically because this has been observed to be about the size of the largest typically observed granulomas. And the feeling is that these are the most important ones to be dealt with. If the infection is contained within a large granuloma, then typically a person will have latent infection. If the largest granuloma leads to dissemination, then we tend to observe active TB. So our model needed to be able to capture the largest possible granulomas as part of its design. Here you can see I've got the laser pointer in case it's not clear. Some of these blue dots are blood vessels, or effectively cross sections of blood vessels, which are randomly located throughout the domain. And the yellow dots are resting macrophages, which also are randomly distributed throughout the domain at the start. And in the center here, you may be able to see our green and red dots. These are the extracellular bacteria. So it's typically been observed that the minimum dose of infection from MTB will be on the order of 10 to 100. So for all of our models, we have initiated the MTB cluster randomly. In this case, it was very central, but this cluster could have been located randomly anywhere within our domain, and always consists of 6 fast-growing and 6 slow-growing bacteria for the same initial conditions in that sense. It should be noted from the name, of course, CompuCell 3D allows for 3-dimensional modeling of biological phenomena, but in our simulations, we've set the length of the third dimension equal to 1, and effectively our model is a 2-dimensional model on a square lattice of size n squared, where n in this case was 500. So our 500 by 500 lattice represents the 2 millimeter by 2 millimeter cross-section of lung tissue. In CompuCell, cells are adjacent lattice sites that have a shared index. So in the diagram on the right, all of these cells that have the index number of 4 all correspond to the same cell, and all of these lattice sites that have the index number 7 will correspond to a different type of cell. The CompuCell model's time step is a Monte Carlo step, and what this means is n squared lattice copy attempts. So each of the lattice sites is cycled through, and for instance, in this case, this lattice site, one of its neighbors is chosen, so this lattice site, and an attempt is made to copy the index number from this site into this site. If the copy succeeds, then that 7 changes to a 4, as you can see here, and if it fails, it remains as a 7. This is done for all of the n squared sites. A sort of destination target cell neighboring the original cell is chosen, and then the copy attempt either succeeds or fails depending on the energy required for it to go through. In our model, a Monte Carlo step represents 24 seconds, and so our model ran for 30,000 Monte Carlo steps to simulate the first 200 hours post-infection. I'll briefly discuss the biological components of our model a little bit further, of course. We've included a range of different cells shown here in the table. We've included 2 phenotypes of extracellular bacteria. Their main difference is the rate at which they replicate, which will depend partially on the local oxygen concentration that they sense. And they can switch between phenotypes if there's not enough or too much or oxygen to stay in their current phenotype. We've included 3 phenotypes of macrophage, which depend on the amount of bacteria that have been phagocytosed at the point in time. So all macrophages start in a resting state. Once they phagocytose any extracellular bacteria, they become infected. Once they phagocytose beyond a certain threshold, they become chronically infected. And if they continue to phagocytose too much more beyond this point, then they burst and they die, and they release the intracellular bacteria back into the domain. Caseum is necrotic dead tissue that is commonly observed by dying macrophages or infected macrophages that have died or have burst. and it's typically a hallmark of TB infection, as caseum is commonly observed in granulomas that form in individuals that have been infected with MTB. The wall structure is an artificial construct we've included in our model to strictly enforce no-flux boundary conditions. We did this because due to the CompuCell framework, if we try to have no-flux boundary conditions without this construct, then some of the cells could occasionally stick to the boundaries and not come free again. So to prevent that from happening, we've inserted these wall cells, which just mean that any other cells that get too close to the boundary will be reflected back into the domain. Some of these cells, the blood vessels, the necrotic tissue, the caseum, and this wall structure will stay static and remain in place throughout time, whereas the other cell types can move and change throughout the simulation. And it's worth noting that any lattice site that isn't occupied by any of these other cell types will then instead be occupied by the medium through which all the cells move through. And we don't explicitly represent intracellular bacteria in our model, but rather for each infected macrophage or chronically infected macrophage, we keep a counter of how many bacteria that they have phagocytosed. And when they then naturally die at the end of their lifespan, or when they burst, then an extracellular bacteria on the quantity of the counter, the number that the counter had reached at the point where the macrophage died, are then initiated back into the domain in a slow-growing state. So if a chronically infected macrophage's counter had reached N, so it phagocytosed N extracellular bacteria at the point it naturally died, then N slow-growing extracellular bacteria would then be reinitiated back into the domain to capture the intracellular bacteria going back into an extracellular state. As discussed, we have two chemical fields in our model, oxygen and a generic chemokine. Both of these evolve over time according to reaction diffusion partial differential equations. And although the diffusion coefficient is constant for the chemokine, the oxygen one is allowed to differ depending on where within the domain the oxygen is diffusing through. So if it senses it's within a granuloma, so for the purpose of the model, we've just defined this as any extracellular bacterium or any immune cell, then its diffusion is reduced, as it's been observed in real-world data that oxygen diffusion through a granuloma is greatly reduced. The oxygen is supplied into the domain via the blood vessels and then consumed by the macrophages and the MTB in varying quantities, depending on which type of cell we're looking at. And the chemokine is secreted by infected and chronically infected macrophages and will eventually decay over time, such that if the infection has gone, then it eventually will decay to zero. I'll now briefly describe in a bit more detail the Glaser-Grainer-Hojeweg model, or the cellular POTS model to give it its other name, how that works in a bit more detail. And then I'll discuss the extracellular processes that we've added in to make our model realistic to TB. So in the Glaser-Grainer-Hojeweg model, for each Monte Carlo step, a new configuration of cells will be proposed. This will be after having gone through each of the n-squared lattice sites and chosen neighboring sites to attempt lattice number index copying attempts for. So for instance, if this cell is trying to copy into this cell, then potentially the red cell will evolve into this shape where this new lattice site has been added to it. Whether or not we accept the new configuration or not will depend on the total energy in the system, which we label H here to stand for Hamiltonian to give it its technical name. And this is just adding up all the total energy in the system that would be required for this new configuration to be accepted. Where in particular, we're looking at the energy for cells to adhere to each other, for cells to maintain their preferred volume constraints, for cells, immune cells to move in response to chemoattractants, and for cells to just randomly move around the domain. So if the energy in the system would be reduced, then we will accept this change in configuration with certainty. If the energy of the system would increase, then it's accepted with an exponentially decreasing probability, where this parameter Tm is just determining the likelihood that we accept energetically unfavorable configurations. Going through each of these energy types in turn, the adhesion energy is calculated by, we sum up the amount of energy needed for contact between one lattice site cell type and a neighboring lattice site cell type, assuming that they both belong to different cells, and we add up over all the lattice sites and all of their neighboring lattice sites. Each cell has a preferred target volume that it likes to be at. So for each step, the difference between that cell's target volume and its current actual volume is calculated and squared times by a scalar. And for all the cell types, we add up all of these quantities to get the total energy needed for cells to be at their current volume rather than their preferred volumes. The amount of energy for chemotactics is only relevant in our model to the macrophages. So the difference in chemotactic energy is calculated as being the difference between the chemoattractant concentration at the source lattice site and at the target lattice site that the lattice index copy attempt has been made into. And all these changes are then added up towards the total energy calculation. And then finally, the random movement energy, again, for our model is just relevant to the macrophages. We don't have the bacteria move through the domain. It's determined by calculating the force required to move the cell, and then this is multiplied by some trigonometric function of the angle between its current direction of travel and the proposed new direction of travel if this new configuration were to be accepted. So that's how the Glaser grain Hoji-Weg aspect works. In addition to this, we have recruitment of resting macrophages taking place every Monte Carlo step. So this blue square here would represent a blood vessel in our model. And we check each of these boundary sites to see if there is a free space. And the number just tells you how many times each site is checked. And so, for instance, for this part of the blood vessel, we check each of these neighboring sites. For this part of the blood vessel, each of these, and so on. So the number indicates how many times each of these boundary sites is checked. If there is a free space, then with some probability, we can recruit a new resting macrophage into the domain, which then on subsequent Monte Carlo steps grows to its preferred target size and then starts to move into the domain. In addition, our cells can change state in any given Monte Carlo step. So for an extracellular bacterium EB, it will check the local oxygen level O. And if this oxygen level is greater than some upper threshold O high, then if it was a slow-growing extracellular bacterium, it will become a fast-growing extracellular bacterium. Otherwise, if the oxygen is below some lower boundary O low, then if it was a fast-growing extracellular bacterium, it becomes a slow-growing extracellular bacterium. And if neither of these is true, whatever state it was in before, it will maintain in this current Monte Carlo step. Similarly, for a macrophage M5, it will check and see if there's any neighboring extracellular bacteria and phagocytose them if there are. If it has phagocytosed anything, then it's yes. If it has not phagocytosed any extracellular bacteria, it stays resting. If the amount it's phagocytosed is greater than or equal to 10 in this case, then it would be chronically infected. If not, it's infected. And if it has phagocytosed the amount required to cause it to burst, which is 20, then it will burst, it will die, and it will become caseum. In addition to this, we also model cell death. So each macrophage is assigned a lifespan. And once it reaches the end of that lifespan, it gets removed from the model. If it was infected or chronically infected, then it turns into caseum and its intracellular bacterial load is released back into the domain. And the bacteria replicate in our model by growing to double their initial volume and then splitting into two new bacteria. And the amount of time it will take them to double in volume will have been determined by their phenotype, at which point a time interval will be set between replication events to occur such that they grow to double the initial volume over the course of that time interval. So that's how the model works. I'll now go into the actual results that we were able to generate using it. The first part of this will be the consistency analysis, which was essentially telling us how many simulations we required to mitigate the model's intrinsic noise and uncertainty resulting from the stochastic nature of the model. We looked in particular at how robust our model outputs were then to some key parameters. And then finally, we generated some actual simulations of the within-host dynamics of MTB infection and compared those to the original WHIDM model that I spoke about from the bonus et al. Paper from 2018 and see if there are any differences between the two models in terms of the outputs they generated. I won't go into too much information on how we conducted the consistency analysis, except to say that the method we've used here was taken from this book chapter by Hamis, Strasiev, and Powerthil from 2021. I've included the reference at the bottom of the slide if you're interested in more information. It was based on this quantity, the A hat value, and the formula showing how it's calculated is shown here. If we have two data distributions B and C of some size n, then if we take a random data point from distribution B, Bi, and we compare it to a data point from distribution C, Cj, then depending on whether the data point from B is bigger than C, the same size, or smaller than the data point from C, this Heaviside functional output 1, 0, minus 1, sorry, 1.5 or 0. We sum over all of these different data point comparisons, and then we can eventually calculate these values, A hat values, which, if it's closer to 0.5, essentially suggests that the two distributions do not have any statistically significant differences between them. And if it's nearer to 1, or the scaled version or the unscaled version could be nearer to 0 or 1, suggests that there are large statistical significant differences between the two distributions, that they probably came from different probability distributions, is the idea. Again, I won't go into too much more detail. If you're interested in the methodology we use, then by all means, I'd recommend the reference shown. The long and short of it is that we found that 300 simulations, as shown by this blue circle here, should be sufficient to mitigate the model's intrinsic noise and stochastic uncertainty. So any results that you see in subsequent sections were generated from 300 repeats of the model. Not shown here, but we also found that 300 repeats is also sufficient for the within-host infectious disease model, WHIDM. So any results for that are also from 300 repeats and averaging. Again, I won't go into any detail on the robustness analysis, except again to refer you to the same reference as before, because the methodology for that was also taken from the same one that we used for the consistency analysis. We investigated the model's robustness to these 4 key parameters, and I'll now go through each of these in turn. The first one we investigated was the bacterium macrophage contact energy. So this was the amount of energy required for a bacterium to adhere to a macrophage, with the idea being that in our model we had phagocytosis would occur if a bacterium and a macrophage adhere to each other. So therefore, if the energy for this to occur was too great and it didn't occur, then phagocytosis would not occur within our model. The calibrated value we first looked at was J equals 10. I would say that don't worry too much about the exact values used, but we compared this to 4 other values of 0, 5, 15, and 20. So the plot on the left shows the statistical significance of the differences between the results generated between J equals 10 and these other values that we considered. And on the right, you can see the total amount of bacteria left at the end of our simulations on average from 300 repeats for each of these parameter values for J, with these box and whisker diagrams. So as can be seen, the total amount of bacteria were broadly consistent irregardless of the parameter value that we used for this contact energy parameter. And the plot on the left suggests there was little statistical significance in any differences between the outputs. For the most part, although there's a small statistical significance for J equals 20 between J equals 20 J equals 10, which made us wonder if we considered larger orders of magnitude for J, would we see larger differences? So we tried a few more experiments, and we did indeed observe that there were then large statistical significance in any differences for larger values of J in our results. As you can see on the right, we would have a lot more bacteria left at the end of simulations if this contact energy parameter was set to be 10 or 100 times larger than the calibrated value. The reason for this, as I touched on, is that if the energy required for adhesion between a bacterium and a macrophage was too great, then phagocytosis would not occur in our model, and therefore the bacteria could replicate and grow large in number without really facing much resistance from the immune response. So beyond a certain order of magnitude, our model was sensitive to the choice of value for this key parameter. The next parameter we looked at was the chemotactic attraction coefficient, which we've labeled lambda chem here. This was just an indication of how much a cell wants to move in the direction of increasing chemoattractant, how much effort it will put into doing so. The calibrated value was 2000, and it was compared to 0, 1000, 3000, and 4000. And you can see the total amount of bacteria left in the end of the simulations on the right. And as can be seen, the amount of bacteria left at the end is broadly consistent, irregardless of the parameter value that we chose here. And the statistical significance of any differences was incredibly small. So it would appear that our model was fairly robust to this choice of parameter value, irregardless of what we set lambda chem equal to. The third parameter we looked at was the dispersion parameter of the von Mises distribution. So to explain what that means, for the random movement aspect of the Glaser-Grainer-Hojeweg model, the angle in the change of direction for the cell was determined by drawing it at random from this von Mises distribution. This is a circular distribution which takes values from minus pi to pi. And depending on the size of the dispersion parameter, you get a very different shape to this distribution. For very low values of what we then refer to as kappa, this dispersion parameter, the distribution looks almost uniform in nature, and therefore any direction is equally likely to be chosen for the change in direction for that Monte Carlo step. Whereas with a larger value of kappa, the distribution starts to more and more closely resemble a very, very steep normal distribution, with the most likely outcome being that the cell doesn't divert from its current direction of travel and keeps going in the same direction it has been before. In our model, the actual calibrated value for kappa was 99. To do this comparison, we've said 99 is essentially 100 and compared that to three other values, up to 3 orders of magnitude smaller and 1 order of magnitude bigger. What we observed is that for 1 order of magnitude in either direction, the differences were not too substantial, not statistically significant. The amount of bacteria left was broadly consistent. But for smaller and smaller values of this dispersion parameter, we would wind up with more and more bacteria remaining at the end of our simulations. Our understanding of why that may be occurring is the feeling that with the way that the lattice copy attempts means that cells move, keeping the direction more and more close to the direction you previously moved in causes effectively the shape of the cell to wind up looking somewhat streamlined, and the cell moves more easily through the domain. Whereas if it keeps changing direction every time step, effectively it almost winds up going in circles and not really moving in any one direction at all. So the actual distance it moves winds up being greatly reduced. And so therefore the bacteria can replicate more quickly before the immune cells actually arrive at the site of infection, as their movement becomes a lot more stop-start compared to cells that more consistently keep moving the direction they had been moving in previous steps. The last parameter we did robustness analysis of was the elasticity coefficient, lambda vol, which is just how much an effort prefers to stay at its target volume rather than switch to some alternative volume. And the calibrated value for this was lambda vol equals 10. We considered up to 2 orders of magnitude smaller and 2 larger. And as you can see, our model was incredibly sensitive to the choice of lambda vol. Specifically, if we went for smaller values of 0.1 or 1, the amount of bacteria completely disappeared by the end of our simulations. Whereas if we set lambda vol equal to 100 or 1,000, we wound up with a lot more bacteria at the end than compared to the value from the calibrated runs. The reason for the bacteria disappearing when lambda vol is too low is the cells have so little effort put in to try and maintain their preferred volume that they wind up essentially disintegrating and disappearing from the model completely. So in theory, this makes it sound like the immune response has done very well, but I should note that we set lambda vol equal to these values for all the cells in a model. So not only were the bacteria fragmenting apart, but so were the immune cells. So essentially, the infection disappears, but so does your immune response. So in other words, we get a very biologically unrealistic outcome with overly low values of lambda vol. Conversely, when we set lambda vol equal to 100 or even more so 1,000, cells had such a strong preference to stay at their preferred current volume that it came at the expense of any movement or anything else effectively. So cells would largely stay frozen in place, just trying to maintain this perfect volume, and as such, the bacteria could freely replicate without the immune cells ever moving towards the site of infection for fear of diverging away from their preferred volume. So yeah, as can be seen, our model was robust to some of these key parameters, but less so to others. To summarize our main calibrated results, we ran 300 repeats for the CompuCell model, and of those, 278 led to what we defined as containment. So in this case, we defined this to be at most 10 extracellular bacteria remaining at the end of simulations, in keeping with the definition from Bonus et al.'s 2018 paper. So it's close to the 90% that I said before. In reality, active disease occurs in 10% plus or minus 5% either way. So certainly these results are fairly closely in keeping with the real world observed proportions. As I said before, if the largest granuloma, which is effectively what we're trying to model, can contain the infection, then typically latent TB will be the end result. So this is largely what we would expect to see in a properly calibrated model, whereas 22 of the 300 simulations saw more than 10 extracellular bacteria left in the end. So in other words, dissemination, and what would probably led to an active disease in such an individual. We also ran 300 repeats on WHIDM. We saw similar proportions, not identical proportions, but all still keeping within the 10% plus or minus 5% of active TB that you would expect. So both of these models have been able to qualitatively agree in terms of how often they lead to containment versus dissemination. They both appear to give a fairly good representation overall of certainly TB infection outcomes in this sense. An interesting finding from this project was that for the first 200 hours, it doesn't appear that spatial organization was important in determining whether or not containment occurred. Our understanding is that this is largely because in the first 200 hours, we do not model antibiotics treatment. Typically, when someone is sick with active TB, the diagnosis does not tend to occur until, at the very least, 1 to 2 weeks after their initial infection. And for some individuals, this can be many months later. So certainly, no one would be on antibiotics within 200 hours of getting infected unless they were extremely fortunate. And in addition, as I said, the T cells do not typically appear at the site of infection until at least 9 days after the initial infection occurred, which is outside these first 200 hours. So unlike the original 2018 paper that looked at the first 500 days or 12,000 hours, which therefore did mean that antibiotics and the adaptive immune response were included, our models, when we did these simulations at least, only looked at the first 200 hours and so did not include these aspects. The reason we only looked at the first 200 hours was due to computational constraints. It was too computationally expensive and took too long to simulate for longer than this period of time. However, these results therefore do suggest that the spatial organization is important in the sense of allowing the antibiotics quick access to the site of infection and the adaptive immune cells quick access. Is less important in terms of recruiting extra macrophages to the site of infection and the initial innate immune response. The next few slides, I'll show the evolution of the cell numbers over the course of the simulations. And this is where we can see that although there was qualitative agreement in terms of the outcomes, containment versus dissemination, quantitatively, how these cell numbers evolved over time were quite different between the two models. So on the left, you can see how the fast-growing extracellular bacteria evolved over the course of the CompuCell model, with the solid line being the mean quantity and the dashed lines being the 95% uncertainty intervals. And we can see that normally the fast-growing extracellular bacteria drop down to almost nothing and only marginally make a small rebound over time in this model. And even at most, we don't ever see more than about 20 fast-growing extracellular bacteria in the domain at any one point in time. In comparison with the WHIDM model from the 2018 paper, the fast-growing extracellular bacteria numbers increase in some cases to hundreds by the end of the simulations, and on average to just under 50 by the end, which is a lot more than what we observed in the CompuCell models. Similarly, for slow-growing extracellular bacteria numbers in the CompuCell model, as we can see, most of the fast-growing ones must have switched to slow-growing as we get this initial jump up to almost 12 for the mean. But then this slowly drops off for the rest of the simulation as most of the bacteria get phagocytosed. Although, as you can see from the uncertainty intervals, we can occasionally have up to 20 left in the domains, which will be the roughly 1 in 10 simulations where dissemination was observed. In the WHIDM model, on the other hand, the numbers drop to almost zero, but then about after the halfway point start to recover in number towards the back end. And in some cases, we had hundreds of slow-growing external bacteria left in the domain after the first 200 hours. The intracellular bacteria numbers are the last ones I'll show. So both models had a similar shape in the sense of this slowly increasing mean amount of bacteria phagocytosed over the course of the model. However, whereas on average in CompuCell, this is about 15 to 20 by the end of the simulations, in the WHIDM model, it was almost 50. And again, in some cases, hundreds of bacteria may have been phagocytosed by the end of simulations. So this was interesting to us. We did wonder why there would be such discrepancies in the actual quantities of bacteria in our model, even though we were seeing the same proportion of containment dissemination outcomes. We had wondered potentially if maybe macrophage recruitment was being handled differently by the models. So we did briefly check to see if the number of macrophages that were left in our model at the end of simulations were broadly consistent. And we found that indeed they were. We started each of our simulations with 105 macrophages. And as you can see, on average, there was about that many macrophages, give or take, in both CompuCell simulations and WHIDM simulations at the end as well. So macrophage, broadly speaking, stayed similar in value throughout. Any that died were, roughly speaking, replaced almost one-to-one, again, within a certain amount of variation. And clearly, the models weren't drastically handling the macrophage recruitment much differently to each other. So all in all, we're left with the interesting outcome that although qualitatively speaking, our simulation results do see similar proportions of containment versus dissemination for the two model types, quantitatively in terms of the amount of bacteria in the domain, we get quite different outcomes. We wonder potentially if this may be due to the nature of the CompuCell framework compared to our WHIDM original, as it's broadly built up on the cellular POTS model approach, which is there looking to minimize the total energy of a configuration of cells. We wonder if potentially that means that CompuCell is then biased towards potentially low energy stable configurations, and potentially there's less possibility for quite large volatility in model outcomes. With tuberculosis, of course, what we observe is that the dynamics can be very volatile. For 1 in every 10 individuals who get sick enough that they require antibiotics, so potentially could have very large bacterial populations build up over time. Whereas for the vast majority of individuals, they will have very few bacteria left, if any. They may not know they ever got sick. They may naturally recover, and they wind up with a latent infection. So inherently in the real world, this is a very volatile disease to model, and therefore possibly if the framework is sort of tending towards these more stable, low energy configurations, this may be harder to capture. We've also observed qualitatively similar but quantitatively different results when comparing CompuCell to other agent-based models in the past. One example I found from the literature was an oncology off-field lattice model presented by Andasari et al. In 2012. Where again the final outcomes were broadly consistent, but the way that the two models got there were quite different. So if anyone's interested, I've left the reference there for further reading. In general, we found the CompuCell framework was a very easy to use framework. The fact that a lot of code snippets could be dragged and dropped into our code from the drop-down menus was very helpful and very intuitive to understand. Certainly anyone who has any prior experience coding in languages like Python would probably find this a very straightforward modeling framework to use, or be able to pick it up very quickly and develop models with it with comparative ease. Of course, the robustness analysis section of this talk suggests that certain key parameters, a lot of which are fairly just fundamental to cellular POTS framework, can be parameters that the model output is highly sensitive to. And some of these parameter values are not necessarily always the most intuitive to parameterize based on real world biological data. It more tends to be arriving at them through sort of consultation with previous models or expert guidance. So further understanding of these may prove beneficial in helping people to parameterize these models accurately. One potential model design aspect that could be problematic, we also thought, is the no flux boundary conditions in both CC3D and in WHIDM. In general, our design of the model to incorporate these no flux boundary conditions may cause issues in the sense that if the initial bacterial cluster starts too near to a boundary, then it would be difficult for a proper granuloma to form around it, as the macrophages can't travel through the boundary to get round to the side of the cluster effectively closest to the boundary. Potential ways that we could address this would be to just always initiate the bacterial cluster centrally within our domain and still randomly distribute the blood vessels and the initial macrophages so that the spatial organization aspect can still be captured, but the granulomas can grow properly, biologically, realistically, and reach the full size that we would expect without these boundary effects coming into play. So to summarize, we believe, to the best of our knowledge, that we have developed the first within-host model of tuberculosis infectious disease dynamics with CompuCell 3D. As to the best of our knowledge, nobody else has used CompuCell 3D for this purpose. What we have found is that although our model is quite sensitive to certain key parameters, all in all, we were able to get very well calibrated results that agrees to a large extent with those produced by WHIDM, at least from a qualitative point of view, even though the quantitatively different amounts of bacteria in the domain were observed. And an interesting finding of this has been that the spatial organization doesn't seem to be important in the first 200 hours post-infection. So therefore, the results from the Bonus et al. 2018 paper imply that it must therefore be more important for the antibiotics to be able to reach the site of infection and for the adaptive immune response to take effect. With that, I'd like to acknowledge and thank the organizers of the 2022 CompuCell 3D workshop. I know that this year's workshop was advertised before my seminar today, and my initial prototype of this model was developed as a part of that workshop, and learning how to use CompuCell 3D was the first part of that week-long workshop. So it was very helpful towards this research. I'd also like to thank the hackathon teammates I had for that hackathon weekend, Jacob and Vasileios, who helped me get a working prototype going by the end of the weekend, although it has to be said, the model changed substantially thereafter in the subsequent years of development. Aminat is a fellow PhD student at the University of Bath, who has also had experience of using WHIDM. So conversations with her were very helpful in making sure that our development of CompuCell 3D model was as close a match to the original model so that the comparisons were valid. And the University of Bath Research Computing Group provided the time and computational resources to conduct the, in the end, thousands of simulations that we needed to gather all the results for this research. So my thanks go to them, to the University of Bath and my PhD supervisors in general for allowing me to do this PhD. And I'd like to thank everyone here for listening. I've added the QR code of our preprint if anyone is interested to read any further. And I'm happy to take any questions you might now have. Thank you.
Okay, time for some questions. I see there were some questions that came up during the seminar. The first one was from Jacob Barak, which said, how much time do these simulations usually take per simulation and on what kind of computer?
The simulations, to the best of my we ran these simulations a few years ago, but I believe it was on the order of about half an hour to an hour to simulate sort of 200 simulated hours at a time. With our sort of supercomputer, you could normally book time for up to 6 hours. So realistically, it was never going to be feasible to simulate the full 500 days or 12,000 hours from the original paper. So we mainly focused on the first 200 hours, as that's when to a large extent, the course of the infection is decided, and you can see whether it will be latent or active disease. And in terms of the computer, it was the Nimbus Cloud supercomputer that the University of Bath provides. I think that's provided through Microsoft Azure, I believe.
And there's a question from Parikshit asking if you change the multiplicity of infection, what would happen? For example, if you started with a single cell bacterial infection particle And would that possibly help you understand more about the contact energy settings?
Well, yeah, we didn't start with a single bacterium, so that's an interesting one. Yes, if I'm honest, I'm not completely sure. Because the original paper from 2018 started with simulations with 6 fast, 6 slow, and one of our primary aims of this was to get an accurate comparison of the two frameworks, and then how closely we could provide that cross validation. We didn't consider other initial multiplicities of infection. It would be interesting future work to consider potentially like a single bacterium and see if the same infection patterns could be observed as it typically they're on the order of 10 to 100 mtb for an infectious dose. So certainly the fact we only considered 12, it would be interesting to see as another potential sensitivity analysis changing that, how that would change the eventual dynamics. But that isn't something that we've done as part of this research yet. So yeah, I'm not entirely sure if the model would have the same results with a different initial MOI.
Okay.
Okay, so yeah, so James, I just kind of wanted to follow up to something you'd mentioned earlier.
Of course.
So it's actually kind of normal that you so when we look at whole host versus single granuloma infections, you can have like up to a dozen granulomas,
Yes.
but if they're all controlling, you're still latent, right? It only takes one active infection for you to like have the like have active disease and be spreading it. And so I think computationally we see something that experimentalists don't, right? It's like a lot of, by and large, a lot of granulomas might sterilize. I think what you're seeing with those initial drop-offs in the bacteria, I think those are real effects. I think there's a lot of granulomas we don't see right experimentally. And so I don't want that to discourage you. So, but I do think that trying to understand, with the single bacterium, like forming infection, I think that would be super cool to show in the paper. I just also just to follow up on another question, I didn't have like I just saw the abstract in the talk and didn't have time to read through the methods, but I think there's kind of different schools of thought and calibration. Like one is to treat your patient infection states as two different states you're trying to calibrate, fit parameters to, versus treating it as a continuum. And I was just trying to understand philosophically how you approach the calibration. Like, did you have sort of these, latent and active specific parameters and common parameters, or how you kind of thought about the process?
So I suppose with regards to the amount of bacteria, like I suppose your first point. I think I guess because our model was aiming to try and replicate the largest granulomas, that's probably why we considered like a larger initial multiplicity of infection compared to, as you say, a lot of the smaller granulomas don't tend to be as impactful in determining whether an individual gets sick or not. So but that's why we haven't necessarily looked at it before. But yeah, I definitely agree. It would be interesting to consider. With regards to the calibration, this essentially as the largest granuloma and the outcome of the large granuloma tends to be linked to whether an individual develops active TB or not. Essentially, my aim with the calibration was to try and get the correct proportions of the outcomes of the model, in the sense that I was aiming for roughly 90% of simulations would have a small amount of bacteria remaining at the end of the runtimes, and 1 in 10, or about 10%, would have this dissemination and a certain amount of bacteria left at the end of simulations. The other aspect, of course, is I was trying to have as close a comparison between the CompuCell framework and the original agent-based model WHIDM as I could. Like in my final thesis, in part, this research was also a good opportunity to introduce the WHIDM model, which was then used towards a later modeling framework as well. So it wasn't a model that I developed, but a good way to introduce it was effectively to say, can we replicate this as closely as we possibly can with CompuCell 3D? So then that model was calibrated to provide the right proportion of 90% of the time there's containment, 10% of the time there's an outbreak. So the parameter tuning that I did, it was, I must admit, largely manual and ad hoc. I'd sort of run some simulations, see the proportions, tweak accordingly, rerun them again. I'd like to say that there was a sort of rigorous algorithm used, but unfortunately I didn't at that time. But yeah, the calibration was done with the intent of getting the model to a point where it could accurately output, most of the time, latent infection, 1 in 10 times active infection or dissemination.
Okay, Kali, I think you had a question.
Yes, great talk, James. I wanted to ask, I know you did the robustness analysis looking at those 4 parameters, but after you got your simulation results, did you do any parameter comparison across the containment and dissemination simulation groups?
Yeah, good question. So the model parameters were left the same for both groups. I guess the thing that changed each time would be, or the main things that we checked, sorry, were the random placement of the initial bacterial cluster relative to the nearest blood vessel, as that had been found to be a statistically significant result in the original 2018 paper, and the amount of blood vessels that were within a certain distance of the initial cluster. Both of those have been found to be statistically significant in determining whether a containment occurred or not. So for each of our simulations, we looked at the average distance and how many blood vessels were within, I believe it was meant to be 0.01 millimeters or 0.1 millimeters. The exact number escapes me. So we got these numbers. We then conducted, I believe it was a 2-sided t-test, and found that the p-value was much greater than 0.05, so concluded that there wasn't statistical significance between in terms of whether containment or dissemination occurred based on this average distance and the number of blood vessels within this distance. So we didn't check anything else beyond that. In theory, like the actual parameters of the model should have been consistent across all the simulations. It was just those random aspects at the start that were meant to be changing, and what we thought would be significant, but that we found was not, which we've essentially concluded is probably because that spatial organization is more important for antibiotics and adaptive immune response. Yeah, it would have been interesting perhaps to have explored a few more aspects. At the time, those were the main results from the original paper that we wanted to explore, though. So.
Thank you. And I did have one more question. For the containment cases, when you looked at the simulation graphical output, did you notice any granuloma-like structures forming with like a core of macrophages?
Yeah, we did. So for the CompuCell models, has the player, I forget what the name is, but had the player showing the simulations occurring in real time. So yeah, we could see that when we were making these model, the calibrated version of the model, we'd see when there was containment, there'd be these, especially due to the chemokine and causing the chemotactic of the immune cells in, we would see these buildups of macrophages and like caseum and dead tissue when there was a containment. Whereas in the case of dissemination, the bacteria just kept spreading more and more radial outward, and the immune cells just couldn't get around it properly. So yeah, we did observe the kind of spatial structure that we would have expected to see, which is very satisfying.
I was going to ask you then, if you were going to extend this model, you've got a question about why you have the divergence between the model that you had previously and the current model. You might do some more fitting and exploration of that. But if you were going to extend it to include additional biological mechanism, what is the next mechanism you would include as you move forward in the study of TB?
I think a good next step, I suppose, would be to add in the adaptive immune response. Certainly our initial hope was to incorporate T cells, and then that would also allow us to have activated macrophages as a different 4th macrophage phenotype. Beyond that, there's other TB models, of course, differentiate the T cells further into the different helper types. Other cytokines have been considered that would help upregulate or downregulate the different immune responses. I think those would have been interesting to incorporate. So obviously computationally expense-wise, it wouldn't probably realistically be possible to run for full 500 days, but I reckon we could potentially have simulated beyond the first 200 hours to say perhaps the first few weeks after infection. And at that point, certainly the adaptive immune response would become a lot more important. So yeah, I think that would be the next logical extension would be to add the adaptive immune response in and see how that impacts some things.
What would the biggest challenges be in beginning to add the adaptive immune response? Clearly, adaptive immune response has many components, so you might not add them all at once. But what would you see the biggest challenges being in adding the next step, and maybe also in calibrating it?
Yeah, I think one thing I found consistently is that the data on some of the biological mechanism for like TB infection dynamics, it feels like there's a lot of data that's not easily or not readily available, or certainly not directly translatable into some of these models. So yeah, I think a big challenge would be trying to parameterize it accurately and try and get the correct responses, especially considering this is a model that's already got a considerable amount of parameters as well. So there's also the issue of trying to identify which parameter values the model is most robust to, if we're adding new aspects in, and therefore which ones to prioritize, trying to get reliable values on. I mean, for some model parameters, we've had to sort of look at how other similar models have parameterized theirs, but of course there isn't necessarily, well, as we've seen here, perfect overlap between, say, how a CompuCell model works and how another similar agent-based model may work. So yeah, I think certainly based on everything I found with this project up to this point, the calibration and parameterization would probably be the most challenging aspects, I think.
There's been a bit of a discussion in the chat about simulation time, and I wondered if you did any profiling of the simulation to identify which parts of the simulation are most time consuming. For example, if you have oxygen, the diffusion constant of oxygen is very fast, so that often makes that an expensive computation. Do you have a sense of which parts of the computation are the most expensive so that you could think about optimizing those?
I think, I mean, it may also be that I maybe didn't write the code the most efficiently. I think I think at the workshop someone pointed out it looked like spaghetti code, so possibly there may be more efficient ways for me to vectorize some of the code in Python. I think there were a lot of for loops through all the different cell types, so potentially that could have been tidied up and written a bit more efficiently. The oxygen may have been possibly one of the slowest points. I think what each Monte Carlo step would be in 24 seconds. I think we then had the diffusion, the chemical fields solved multiple times per Monte Carlo step in order to keep them accurate, because otherwise it would have meant shortening the Monte Carlo step down. And it could have potentially become a lot more complex. I think as a first step, it would make sense for me to go back and rewrite the code more efficiently. Certainly, I think would be the first step.
I'm looking at this. I'm pretty sure, I have to look into more details, but I'm pretty sure you can accelerate it significantly. This can be, my gut feeling is that you can times 10 or even more do it if I'm right.
Thank you, Jacob. Okay, I want to thank you for coming, everyone. And I want to thank you, James, for giving a presentation today. Really nice to see how that work has progressed. Great job. And maybe we'll see James come back in a year or two and present new work. It doesn't have to be, of course, on CompuCell. We'd be happy to hear about the other kinds of modeling you're doing as well. And other people from your group, we'd love to hear from as well. So please don't be shy about saying you want to speak here. And we're always happy to give people an opportunity to explore their research. Thank you, everybody, for coming, and we will see you in a few weeks. Bye.